Bio, welcome back. Today we are going to be learning about the Hardy-Weinberg equation, so get excited. This equation can be used to test whether a population is evolving, and today we're going to learn about the variables in the equation and what they're used for, and then in the next video we'll be doing some math. So make sure you like, comment, and subscribe so that you're notified when my next video comes out. Genetic variation, as you know, is required for a population to evolve, but just because there's genetic variation doesn't mean that it will. So there's a few other factors that have to happen in order for evolution to occur, and we'll examine those in this video as well. A couple vocab words that I took from Khan Academy. Microevolution is the change of alleles that are occurring over a short period of time, and that's what we'll be mostly studying in chapter 21. And that goes along with population genetics. So this is the change in allele frequencies in a population over time. And then finally, the allele frequency is going to be referring to how common a particular allele is in the population. And then the gene pool is all of the copies of the gene in that population. So one important distinction is to understand the difference between microevolution and macroevolution. You've probably studied a lot about macroevolution, which is the evolution involving a large change that maybe ultimately results in the formation of a new species. But we can also have microevolution happen, which is the change in a small scale of allele frequencies in a population. The population is a localized group of individuals who can interbreed and produce fertile offspring. They are not necessarily isolated from one another geographically. So, for example, when we talk about humans that are in the population of humans in LA, we can breed with humans that live in other populations, but our population, they put a geographic timestamp on it just to kind of discuss that particular population. This is, and especially because you most likely would breed with other humans in LA. You would most likely breed with the geographic location closest to you. Populations are the smallest unit that can evolve. So we talked about, in the past videos, how evolution cannot really happen to an individual. It's more the accumulation of change over time that causes a bigger group, such as a population, to evolve. So the gene pool consists of all the alleles for loci in a particular population. And this is basically the whole set of alleles that exist in a population. The alleles, if you remember from back in our unit on heredity, those are basically the different versions of a gene. So, for example, we have a big W, which could represent the dominant purple allele, and a little w that represents the recessive white allele. So the big W and the little w would be two different alleles in our gene pool. And then the allele frequency tells us how often a particular allele appears in the population. So how often are we going to see a big W, or how often will we see a little w? This is different than the genotype and the phenotype, because the genotype is asking about the pairs of alleles, and the phenotype is asking what the physical ramifications of having those allele pairs, those genotypes, are. We can calculate the frequency of an allele in a population, and for diploid organisms, which most are that we'll be dealing with, the total number of alleles they can have is 2. And then the total number of dominant alleles is 2 for homozygous dominant and 1 for homozygous recessive, same for recessive alleles. So if we wanted to know the allele frequency, we would see we have 1, 2, 3 out of 6 that are big W, and then 1, 2, 3 out of 6 that are small w. If we have two alleles at a particular gene locus, we use P and Q to represent their frequencies. P usually represents the dominant allele, and Q represents the recessive allele. And the frequency of all the alleles in a population will add up to 1, because that would mean 100% of the alleles. So for example, P plus Q equals 1. This is an example of how we would calculate the allele frequencies. So to calculate the allele frequencies, we want to divide the number of copies of each allele by the total number of copies. So if we look at this picture, we can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 of big W allele, and then 1, 2, 3, 4, 5 of little w allele. And the total number is 18. Since we talked already, we represent them with P and Q. P is usually used to represent the frequency of the dominant allele, so big W. 13 over 18 gives us 72%. Q is the frequency of the recessive allele. 5 over 18 equals 0.28 equals 28%. And when we add those up together, we're going to get 1 if we add the decimal version, or 100% if we add the percentages. So P plus Q still equals 1. That means that if you have P, you can find Q, and if you have Q, you can find P, as long as you have 1. It's also important to note that these are the frequencies of the allele itself. So remember, not the genotype big W, but just how many big W's there are. So when we talk about the genotype frequency, that is a little bit different, because now we're talking about the pairs of alleles. So we have big W would be contributing to this one. So we've got 1, 2, 3, 4, 5, 6 out of 9 offspring. For this frequency, we have 1 out of 9, and this frequency, we have 2 out of 9. And we can see those here. Phenotype frequency is also different, how often we see purple versus white. But then the allele frequency is how often we see each of these alleles. The allele frequencies, when they change, that indicates the population is evolving. So maybe we have one original generation here, and we've got 0.72 or 72% RP and 28% RQ. But after many years, we take it again and we see 0.44, so 44% RP and 56% RQ. We might see a little bit of a difference. So we'll stop there, and in the next video, which is also on this section, we're going to learn how to apply this to the Hardy-Weinberg equation.